Hello, my name is Milad Golkaram, a PhD student of Professor Katja Loos in the University of Groningen. I'm glad to present the study done by our research group, including me, Professor Loos, Dr. Fodor from University of Groningen, and Professor van Ruijmbeek from University of Leuven. The title of this work is Linear Viscoelasticity of Weakly Hydrogen-Bonded Polymers Near and Below the Solder Transition. Studying the linear rheology can give insights into the dynamics of polymeric material, especially if the polymers consist of additional interaction such as hydrogen bonding. The effect can be interesting, as there will be an interplay of association kinetic versus polymer chain relaxation. However, the effect of weak hydrogen bonding introduced as side groups can be investigated using theories of gelation and the corresponding scaling. This effect can be resolved when there is no dispersity in chain lengths and sticker number density. And a flexible sticker has to be used between the sticker and the main chain. Therefore, a synthetic approach was used benefiting from raft polymerization to make different copolymer compositions containing 0, 10, 30, and 100% thymine along the backbone. In the case of 100% sticker, namely PTHY100, no dispersity exists, and around 159 sticker was calculated via proton NMR spectroscopy. According to the theory evolved by Chen and his co-workers, a transient gel can show scalings in linear viscoelastic regime. Depending on the extent of gelation starting from -1 for a linear polymer, different regimes can be seen with their specific slope in the log-log scale. If the extent of gelation is less than Ginsberg point, the cluster will relax using mean field percolation theory. Therefore, in the gelation extents below Ginsberg point showed by the red line, for high frequency, Rouse relaxation is expected. In the intermediate frequency, a slope of 1, and at low frequency, a slope of 2 regarding Maxwell relaxation is anticipated for Struge modulus. With increasing the gelation extent, we enter a new regime which can relax via mean field and then critical percolation theory into intermediate frequency. The low and high frequency parts of the relaxation spectra is the same in all cases. Eventually, the extent of gelation will enter a regime which was introduced by Professor Rubinstein. In this regime, there is a competition between the time scale for the dissociation of the cluster into two clusters and a relaxation of the original cluster. This time is called the effective break time. Therefore, we managed to apply the time temperature superposition to compare our results to the theory. However, a thermorheologically complex behavior was observed. In order to overcome this issue, a pseudo-master curve construction was proposed using the G' data, since according to the Maxwell equation, the G' data are more sensitive to the sticker dissociation or slower modes of relaxation. Nevertheless, from this master curve, it can be seen that higher temperatures, 75, 85, and 90 degrees C, the terminal relaxation for sample PTHY100 becomes visible, and the relaxation time, which is controlled by sticker lifetime, shifts to higher frequencies with temperature increase. This indicates an acceleration of the sticker's dissociation in comparison to the Rouse type motion. Therefore, a pseudo-master curve could be compared with the theory, implying that with increasing the sticker composition, a transition from linear chain to cluster is happening. For the case of polymethyl acrylate, it has been argued that the appearance of a terminal slope smaller than 1 and 2 is due to phase separation of the acrylic acid groups formed by accidental hydrolysis during the synthesis of polybutyl acrylate. However, the presence of non-domains and non-phase separation of poly-N-alkyl acrylate is well known for the side chain incompatibility. In these four master curves, we clearly see that whenever the incompatible side chain is absent, the shoulder is also absent. In the case of PTHY100, we see this, and even the spacer between the thymine does not phase separate due to the bulky thymine group. Therefore, to sum it up, two conclusions were obtained. First, the formed clusters were stable to very low frequency or longer time scales. Therefore, weakly associated networks do not dissociate rapidly and can relax as a cluster before the dissociation of the sticker can lead to the relaxation of the linear precursors. This in fact means slow kinetics. Conclusion number 2 is that the formation of the gel was not possible even with a fully functionalized polymer bearing 159 stickers. Therefore, the weakness of the sticker is their inability for complete association. This, in fact, means that thermodynamically the association is not favored. At the end, I would like to acknowledge the following people: Professor Quan Chen, Dr. Salvatore Costanzo, and Dr. Giuseppe Portale for the exciting discussions. And of course, the NWO for the financial support.